Good day. I am Dr.K Gireesh, physician, neurophysician and neurosurgeon. This is the RNA series 25A. RNA is nothing but random questions in neurology and neurosurgery with answers given by Dr. K Gireesh, and that's myself. This is to benefit the practitioners of neurosciences, neurology and neurosurgery, and everyone in the medical field, and this is for a lifelong learning and also for the public awareness, and that is RNA series, this 25A. The question that I am addressing in this series, RNA series, is what is ischemic penumbra and what is the clinical relevance? The definition as coined by Fisher, which is widely accepted, it is the area that is a ischemic tissue potentially destined for infarction but isn't irreversibly injured and is therefore the target of any acute therapies. So, if this is the area of infarction, there is an area surrounding it which is not dead but it can go in die very soon. That is the ischemic penumbra. The original definition of the penumbra referred to areas of the brain that were damaged but not yet dead and offer promise to rescue the brain tissue with appropriate therapy. Now, the penumbra, this is the infarcted region, this is the penumbra that is surrounding it. The penumbra region typically occurs when the blood flow drops below 20 ml per 100 gram per minute. At this point, electrical communication between neurons fail to exist. Cells in this region, this surrounding region, are alive, but metabolic pumps are inhibited. Oxidative metabolism is reduced, but neurons may begin to depolarize again. So, these areas can be revived. Areas of the brain generally do not become infarcted until the blood flow to the region drops below 10 ml per 100 gram per minute. So, this is the zone of infarction. This you find an area surrounding it, and here the blood flow is below 20 ml per 100 gram per minute, but above 10 ml per 100 gram per minute. When it goes below 10 ml per 100 gram per minute, all the cells also will become dead. So, this is very, very important. When it goes below 10 ml per 100 gram per minute, the glutamate release becomes unregulated, ion pumps are inhibited, and adenosine triphosphate synthesis also stops, which ultimately leads to the disruption of the intracellular process and results in neuronal death. So, infarcted area, surrounding this area is the ischemic penumbra, where the blood flow is below 20 ml per 100 gram per minute, but above 10 ml per 100 gram per minute. So, cells are just surviving. So, this is the area of cells that surround the infarction, which we are trying to save by giving thrombolytic drugs. The MRI, we can do both, the magnetic resonance imaging can in fact estimate the size of the penumbra with a combination of two MRI sequences, perfusion weighted imaging and diffusion weighted imaging. Diffusion weighted imaging will see the infarcted core. Perfusion weighted imaging shows decreased blood perfusion both in infarcted core and the penumbra. So, that is the diffusion weighted and the perfusion weighted. Suppose the diffusion weighted is as the size of the perfusion weighted, that means diffusion, perfusion, there is matching. That means you cannot give a thrombolytic drug. But if you find the diffusion is weighting, the infarcted core is less, but the penumbra is more, as seen in the perfusion weighted imaging, that means diffusion perfusion mismatch. In that case, you can give a thrombolytic drug. So, this is very, very important. The magnetic resonance imaging can estimate the size of the penumbra with a combination of two MRI sequences. Perfusion weighted imaging shows decreased blood perfusion in the infarcted core and the penumbra. Diffusion weighted imaging can estimate the size of the infarcted core. Both of these sequences somewhat overestimate the volumes of interest, but the size of the penumbra can roughly be estimated by subtracting the abnormal volume by the diffusion weighted imaging from the abnormal volume of the perfusion weighted imaging. The penumbral area can also be detected based on integration of three factors, and these factors include the site of vessel occlusion, the extent of oligemia, that is hypoperfused area surrounding the penumbra but not at risk for infarction. At that moment, and the mismatch between this perfusion defect and the area of the brain already infarcted. A higher volume of penumbra around the cerebral infarction means a greater volume of potentially salvageable brain matter by thrombolysis and thrombectomy. So, a larger perfusion weighted area or larger ischemic penumbra, that means you can have a larger tissue and they will be salvageable if you give a thrombolytic drug, and those neurons in this particular penumbra can be saved. Such therapies have a greater effect on the regaining the function, such as a movement after a cerebral infarction. After the initial ischemic event, the penumbra transitions from a tissue remodeling characterized by damage to a remodeling characterized by repair. In the penumbra, very important, this is the infarcted zone, and in the penumbra, microglia are thought to exert neuroprotective effects via specialized contacts with neuronal somata, termed somatic junctions. Understanding and supporting these microglial actions could broaden the therapeutic window and lead to a higher amount of preserved nervous tissue. Now, this is very important. So, these are all given various publications by Henhall, then A New Penumbra, and published in Nature Medicine, published articles in Stroke Journal, and so forth. Now, how did this concept of penumbra evolve over the history? The concept of the ischemic penumbra was developed in Lindsay Simon's laboratory, the National Hospital, Queen's Square, London, in 1976, by combined focal measurements of neurofunction, blood flow, and extracellular potassium in the baboon brain following an MCA occlusion. Critical levels of blood flow was observed for function and energy metabolism. These results and the first mentioning of the term ischemic penumbra were published in 1977 in the Stroke Journal by Fisher et al. With titled Current Concepts of the Ischemic Penumbra, Stroke 32, 11th supplement, page 2657 to 2658. The first decade of research focused on the physiological profile of the penumbra tissue after stroke, mapping the cerebral blood flow and quantifying the oxygen and glucose consumption to define these areas. The second decade revealed the mechanism of the neuronal cell death. As the biochemical pathways were dissected, penumbral science became a rapidly evolving area of molecular biology. The third decade of penumbral research found a transitional leap as using the PET scanning, which can identify the brain tissue with the decreasing blood flow, and the MRI has the ability to detect portions of ischemic tissue that has not yet died. These images have allowed vision into the brain to see the area of tissue that may be salvaged, and that is the penumbra. Infarcted tissue and the surrounding cells surrounding that infarcted tissue is the penumbra, where the cells are not dead, and these are the cells that can be salvaged or can go in for recovery, wherein you give a thrombolytic drug, very, very important. So, that is about the ischemic penumbra. Thank you very much.